Hello, and welcome to our webinar on continuous manufacturing, hosted by Just-Evotec Biologics. Our speakers are Brian Kelley, SVP Process and Product Development at Biotechnology. Vir is addressing some of the world's most challenging infectious diseases using a portfolio of biologic modalities in both the developed and developing worlds. Formerly, Brian was Vice President of Bioprocess Development at Genentech for 10 years. He joined Genentech after 15 years at Genetics Institute Wyeth. Brian obtained his BS in Chemical Engineering from the University of Wisconsin-Madison and his PhD from MIT. He is a member of the National Academy of Engineering and has been recognized by the ACS Biotechnology Division with the Michaels Award. And Randal Bass, EVP Process Design and Biotherapeutic Operations at Just-Evotec Biologics. Randal is responsible for upstream, downstream, analytical development activities, and J-Pod manufacturing operations. He began his 20-plus year career in the industry at Amgen, leading analytical and cross-functional process teams. Most recently, Randal served as a director of process development, overseeing the Washington Site Analytical Sciences function. Randal has broad experience developing large molecule therapeutics, both novel and biosimilar, from early stage first in human to commercialization through post-launch support and second-generation commercial processes. He has been an author of numerous regulatory filings to the FDA, EMA, PMDA, and many other agencies. Brian and Randal discuss the questions, how real is continuous manufacturing and what are the challenges? And now, ladies and gentlemen, enjoy our presentation.
Hello, everybody. Welcome to the call. Good morning, Randal. Nice haircut, I would say. I wish I could afford a barber, but times are hard down here in San Francisco. Randal and I thought we would kick off the session today by kind of viewing where large-scale CHO manufacturing has gone in the last 30 years with the manufacturing of recombinant antibodies. And so the comparator, the natural comparator to a continuous manufacturing process is a current fed batch CHO culture at a number of scales, depending upon the demand for your product. But I'm going to look at very large scale manufacturing here in particular, because if one considers a driver of either capacity to produce or cost of goods, dollars per gram of antibody manufactured, it's a very large scale manufacturing facility that needs to be the comparator. And so I had the pleasure of working at Genentech for 10 years, had nothing to do with the early days of scale up of CHO cultures, but kind of became aware of that history more intimately when I joined Genentech in 2007. So we'll begin with very large scale cell cultures first being established in Genentech's South San Francisco facility, where 12,000 liter stainless steel bioreactors were used for fed batch production culture of initially TPA in 1987, the first recombinant protein licensed from recombinant CHO cells, then DNase, and both of those are fairly complicated molecules with regards to either their post-translational modifications and glycosylation and fairly low expression levels. Then Genentech pivoted into chimeric and then humanized antibodies. Rituxan was the first recombinant antibody, OKT3 was not recombinant, in 97. And then Herceptin and Avastin, I list them because they were sort of three blockbuster antibodies from the 90s and very early 2000s. They were manufactured in Vacaville, a town near Sacramento in California, that had a 12K reactor panel, 12 bioreactors in a purification train, and has later been expanded to an even larger second plant we'll talk about in just a second. So back in the day, mid to late 80s, early 90s, titers were hundreds of milligrams per mil. We made ourselves feel important when we said, wow, that's like 800 milligrams per mil. Today, that would be 0.8 grams per liter, and we'd be kind of embarrassed. But there were bragging rights back then going up to maybe 2 grams per liter. Now high titer production cultures greater than 4 grams per liter cause purification bottlenecks in older facilities. So in the mid to late 90s, I can't count how many conferences I went to where I heard the sky was falling because of the purification bottleneck. Well, it wasn't because the unit operations weren't capable of purifying larger titers, higher titers, or larger batches. It's just that the plants were designed for smaller titer. And so once you build a new plant and you put in just simply a bigger in-process pool tank chain to capture or hold material between steps, the purification bottleneck more or less went away. It was merely equipment limitations. It wasn't really a need for different unit operations. So that kind of enabled what is current state. Bioreactor volumes as large as 25,000 liters, a period where multiple companies and some CDMOs built 6-pack design facilities where 6 by 15, 6 by 16,000 liter reactors were a common design basis. Today, the largest plants are 8 by 25 or 12 by 15K. So that's 200,000 liter or 180,000 liter. And they've all been designed for very large scale purification downstream. So now we have a very different situation with regards to the total mass that can be accommodated in a single manufacturing batch. I will compliment all the cell culture biologists, molecular biologists, and engineers who have worked so hard to develop high titer processes. For fed batch, we hit 10 grams per liter, as I recall, back in Andover when I was there, circa 2005. And today it's not that common, but it's still noted that a number of companies or CDMOs can accumulate 10 grams per liter in a fed batch process. And it's by optimization of the CHO cell line, the media, the promoters, and the means of integrating the genes for production of the antibody. And if we think about it, at the largest scale of production, that could yield 180 kilograms in a single batch. It's kind of more like insulin than it is like a recombinant protein. I mean, that's a huge, huge batch potential. And I list sort of here uncited work. Several companies have reported that they have developed processes and run at scale that produced over 100 kilograms per batch. And so now I close with, before I ping pong over to Randal, several CDMOs, at least 4, and 5 is on the cusp of, I don't know, Celltrion's capacity, have developed very large scale capacity. They're building these big plants. They continue to expand. They are co-locating drug product with the drug substance manufacturing. That's a huge advantage, in my view, for not needing to ship thousands of liters of frozen drug substance around the globe. And I close with the final bullet. A single 180,000 liter plant at 10 grams a liter and 70% yield would produce, if it were fully occupied and was blasting through the production schedule that year, over 30 tons of protein from one plant. And so Samsung has 5 plants. Fujifilm has 3 in Denmark and are building 2 more in North Carolina. Lonza has Visp. So there's an enormous production capacity, and I'm wrapping back to the top. Enormous production capacity available, and that could enable probably the lowest cost of goods possible by virtue of economy of scale. And so there's no capacity limitation, and there's an attractive cost structure for a manufacturing. So with that, then over to the Bass Man.
Well, first, I want to go on record in saying that Brian Kelley is largely reviled by his industry peers, myself included. No, I'm just kidding. Thanks for doing this, Brian. I think part of the reason I asked Brian to talk about Vacaville because I drove through Vacaville about a month and a half ago, and it reminded me of what an amazing success story it was. Having grown up in the industry at a time when we actually were pretty capacity limited, Vacaville answered the question of how are we going to produce these things. And the important part is talking about Rituxan, Herceptin, and Avastin, and I worked at Amgen, I worked a lot on Enbrel. I mean, these drugs are miracles. And we did anything we could at that time to try to produce the drugs to make sure that we got as many patients served as possible. And regardless of where we end up on the, we're leaning more towards continuous and small intensified. It's really about, Brian hit on it, it's really about making sure that these things are cost effective, that we can get them to patients, because patients actually deserve our help. When I go out in the industry and talk about this, there's not a real consensus on what it is. So I'm going to just tell you what our flavor is and the way that we're approaching continuous manufacturing. So what is continuous biomanufacturing? What does it look like? I mean, literally, because I'm going to show you some pictures. From our standpoint, there are some advantages in terms of the mass output that we can get and the cost of goods manufactured, or COGMs, that we can get, because there's some caveats towards scaling up. And full disclosure, that was the first 15, 18 years of my life too. Is all I did was work on processes to make initial clinical batches and then go transfer to a commercial facility and scale up to exactly the types of facilities Brian's talking about. Very large fed batch stainless reactor designs with big fixed purification trains and hold up tanks. So we've gone in a little bit different direction. We can also talk about what does continuous manufacturing get you from a facility. But just by way of quick context, what is this thing that we call Just, and how did I get on this road to continuous manufacturing? It actually started before we ever started Just. We actually started thinking about intensification and single-use bioreactor systems and smaller facility designs back at Amgen. So credit to where credit's due, Amgen supported us in this back starting before 2010. But we started Just in 2014 with a bunch of people that had worked on that. In 2019, we merged with Evotec to create Just-Evotec Biologics. And that was really around operationalizing the technology that we've been building. 2021, we actually built our first J-Pod facility. And so J-Pod facility, it's open, it's in Redmond. It basically takes the technology that we created and built our first small footprint continuous manufacturing facility. So this is actually real. Hadn't even finished the commissioning, and we started designing and building the second J-Pod facility, which is in Toulouse, France. So we're actually in the process of building that facility now. It should go live, well, it is going to go live in 2024 with our commissioning batches. This year earlier, we had a big validation of our technology platform where we announced a strategic partnership with Sandoz to bring that technology to bear on their biosimilars. It's all about how we can affect rapid development and low cost of goods manufacturing. So that's kind of the story of where we got to. I can show you what it is, but tying back to another thing that Brian said that was really important, we actually had to design all this stuff to work together. So it's not just about building a facility. We actually have to build the technology that goes into the facility. And I'm with Brian on this one, which is hats off to the molecular biologists and cellular physiologists that have engineered these amazing cell lines and vector systems for high productivity. In our world, we call it J-design, but it's really about everything, about a discovery library that's based on a humanoid antibody library, so you can build better molecules through what we call J-molecular design. So if you get the molecule right, like if I asked Brian if he would rather make Rituxan or Avastin, I'm pretty sure he's going to say Avastin. It's a much better, more modern protein sequence, and it's actually easier to create. We had to build everything, cells, vectors. To get continuous to work, you actually need to have a highly intensified small scale system. There's a schematic of that. And that's actually all so that it fits in what we call a J-pod. So we actually used the pods, which are autonomous clean roots, as a forcing function. So we knew if we were going to try to reduce costs, we actually had to reduce the scale of the facilities down to what I call the human scale of manufacturing, meaning a human can actually move things around in the facility. There's nothing hard piped in it other than like a water for injection, the clean water systems that we use. Everything's flexible, everything's on wheels, everything's small. But to do that, We had to engineer everything so it fits together and it actually can fit into that footprint. So this is kind of a complicated view of what our version of continuous end-to-end manufacturing is. And so this is sort of a fish lens of going through the facility that I'm in right now, which is in downtown Seattle. It's our clinical scale manufacturing facility pilot plant. So it operates at 500 liter scale. Our commercial facility doesn't get much bigger than this. It actually operates at 1,000 liters. But this is all of the equipment in an engineering run actually in use. So a 500 liter perfused bioreactor, sort of on the schematic sweeping from left to right, that intensified reactor is physically connected to a continuous protein A capture system. So this is one of the ways that we got around some of the scaling issues that Brian talked about. Of when we went really big and titer started getting good, you start getting to a point where it's difficult to process the mass. One way of doing it in fed batch is just create bigger tanks. You have bigger columns, bigger hold tanks, and you just kind of brute force your way through it. The way that we actually went through it is we actually designed the system to operate in a continuous mode. So you can use time as a lever. So instead of like collecting everything all at once and processing everything, we actually get the bioreactors up into a stable state. And then we just constantly collect the product and forward process it. So what's interesting, if you look carefully in this picture, every single one of these unit operations is in fluid connection with all of them. So it literally means there's tubing connecting every single one of these unit operations going from the reactor through capture, low pH viral inactivation, continuous filtration, and then a couple of flow through and frontal chromatographies that make this all possible. So we can go through all of the polishing steps and then through a continuous viral filtration, and then finally into a continuous UFD effort, inline concentration, inline diafiltration. So this whole thing actually runs continuously, and it's pretty small. Like I said, the reactors only get up to 500 and 1,000 liters. The unit operations themselves are actually fairly traditional. This unit op flow diagram that I have is fairly common in our industry, but it just runs continuously for weeks. And so that's what the advantage here is that once you get it all to work and you get the cells and the vectors engineered and high productivities, and when we talk about productivities in this format, we don't talk about grams per liter, we talk about grams per liter per day, because you have to use that time component. So it's small and it's incredibly productive. So this engineering batch is producing, I think this one was producing 8 or 10 kilos in a 18-day process. So again, time's your ally here. So this is what it looks like. When we go into commercial manufacturing, then we do all the normal segregations that you would normally have, meaning cell culture and then continuous capture. We go through a purification pod, and of course we go through a post-viral. We still have to do post-viral. So again, all in fluid connection, 100% disposable from vial crack to drug substance fill. All of these things are single-use systems that are now common in our industry. So I'll talk about some of those advantages when we get into the roundtable. So this is one version of continuous manufacturing. Some things that it gets you is on the next slide that I'll talk about really briefly. A lot of productivity, and again, time becomes a component. So in that diagram, or sorry, that graph, you can imagine that just by changing the duration of your runs using the same equipment at the same scale with the same unit operations, if you just started off early on, and this is actually how we develop our processes, we leverage our platform and really, we only have to produce a few kilos for early clinical trials, but that same process can be adapted very readily without the need for scale up to commercial manufacturing, where we can make tens of kilos, 30, 40 kilo batches out of exactly the same process using the same process equipment. We do a lot of cost modeling. When we use time as a component and fully utilize our facilities, they're actually pretty cost competitive. Our commercial COGS can range to as low as $50 per gram. The advantages there are, again, we can scale out, we can replicate these things because they're all single-use systems. They're small, they can just be wheeled in. And we think that we can actually achieve really low and competitive COGS relative to traditional CDMOs. And we could talk about the caveats to that in just a few minutes. So again, some advantages, we don't have to scale up. We can actually move from product to product very quickly because we can change the facilities out and change all of the disposables in a matter of hours as opposed to many days. Another advantage, they're fast to build. These things go up quick because the pods can be constructed at the same time we're doing cores and shells. And pods are actually are getting commonly used throughout our industry, it means you can build some of the most critical parts of your infrastructure, meaning your clean rooms, at the same time you're building your facilities. We've done that here in Redmond, which is just about 20 miles away from where I'm sitting. And we're doing it again in Toulouse, where these things come up very rapidly. From the time we're scraping dirt until we're into our commissioning runs is about 18 months. So they're very fast relative to traditional plants. Traditional plants take a lot longer to build. I would argue that on the order of roughly 3 or 4 years to build them, because they're physically big. They're engineering marvels, and that's actually how those facilities operate. Is that they're incredibly complicated because they're all physically connected using hard piping, because of the volumes and the scale of things that you have to operate at. They're also really expensive. They work great once you've built them, don't get me wrong, but the last one that I built in my career was part of a build. It was basically scraping a hole in the ground and dumping $1 billion into it. So they're very expensive. The J-Pod facilities are a lot cheaper. They're very simple facilities. So they cost about a quarter of the price. The facilities themselves are more accessible to people and companies that are starting up like us that don't have $1 billion to drop into the ground. So another advantage that we see is that when we do the modeling for carbon footprint and environmental sustainability, even with disposables, because they're so much smaller and you don't have all of the cleaning and sterilization required in the big fed batch facilities, they actually use a lot less natural gas and they use a lot less water. And they still produce at the commercial scale. The one in Redmond, we can show that it can produce over 2 metric tons of drug substance per year.
Thank you very much, Brian and Randal, for your great presentation. This is the end of our event. Thank you very much for your participation. If you would like to get in touch with our speakers, please email us at info@evotech.com.